Hi, welcome back to the next episode of the Keto Naturopath podcast on YouTube. So today I'm going to give you some homework assignments that I hope you'll follow up in, because I do believe it will change the course of your life. Pretty big claim, isn't it? You know, a little hyperbole is good. It's not hyperbole. I would like you to read two articles, and I'll put the links to these two articles to, actually neither one is a study. They're basically reviews, and I find them very exciting. One is going to be a little more complicated than the other, so I'll help you there. But the two studies are this: The Brief History of Choline by Dr. Stephen Seisel from the University of North Carolina. And he's pretty much, the last 20 or 30 years, the major researcher into choline in its different applications. Choline research and the applications to human health started a long time ago. He writes this paper. He's done a lot of research and a lot of really technical research about the SNPs relative to choline and premenopausal, postmenopausal women, and even men in certain SNPs leading to infertility. Another topic, another time. But let me, put this out there in terms of, if you can go through this, and maybe it's, not even, if you enlarge the print like I do sometimes, you have maybe 5 pages to read. And if you want to go through all the references, which are equally educational and interesting, I would say it's the beginning of the novel of choline and why it is so acutely important for you. So it basically talks by when do we really think we discovered choline and what was it, from 1850. So it's not that long ago. When you think about, it's really about pre-Civil War, Civil War time, and this was, of course, in Europe. It wasn't in this country. In Paris is where the story starts, but I'm not going to give it to you chapter by chapter, but I will outline some things here by saying that there is a sequence of discoveries that are really worth knowing. And so I'm going to give you sort of the highlights, but I really do hope you go off and you get this link and you download it and you print it out. And what you can do is you can print out just the first 4 or 5 pages of the article and not do the references if you don't want, and read that. That should take you all of a half hour. If you get engrossed, if you're like me, you're going to slow down as you read and want to understand each sentence as it's sort of strewn out in front of you, because it's such a powerful story. And this will change your life, because choline is not just another nutrient. It's not that, we just discovered folate. And what is folate for? Or B12. You could say the last 100 years is all about discovering nutrients, which is pretty much true since the early 1900s. One after the other, we found out what was vital, primarily because there was a population of people that incurred a deficiency of a certain thing, and it had a certain sort of disease presentation. And because they had the presentation, it had to be investigated, and we found out, it led back to their diet. Pellagra is actually a really interesting story that I've covered before on podcasts, a couple of podcasts, about how corn was not appropriately cooked in the United States. That corn, just as an example, corn has to be bathed in an acid lye, which is basically fire ash. It's stirred around, and it needs to be extracted, stewed, if you will, in this lye, and then extracted, and then made into the tortillas and so on and so forth that are famous for Mexican food. Well, when Columbus discovered it in 1492 or 3 or 4, he forgot to bring to Europe that piece of, this is how you use corn. He said, corn, look we saw people eating it on the cob, we saw eating it in so many different ways. So for 400 years, there was this thing called pellagra, which is a vitamin deficiency caused by corn. Who would have thought? I'm not going to spoil the story, and I hope you will go look into that one, or at the very least go back and listen to those podcasts. But the point there was, taking something out of context could be very dangerous for people, and it usually has to do with their diet. And most nutritional deficiencies, maybe I should say all nutritional deficiencies, really start with diet, and something is being left out, something is being omitted and or blocked, if you will. Now we're in the era of the last 70 years, certainly since World War II, of progressively more and more convenient food, more and more processed food, more and more food that not only has things removed from it, right? Think of refined carbohydrates to complex carbohydrates to refined carbohydrates. More and more nutrients are being shed along the way. And then into that, there's another layer of preservatives and all the other things that have to be added to give it shelf life and color and so on and so forth. So the food that we have now, food in quotes, is nothing like our great-grandparents had or our great-great-grandparents had. And we are in a different world. We are in a modern world of self-induced, culturally self-induced nutrient deficiencies. They also call it hypernutrition, but it's just a play on words. The reality is we are nutrient deficient in many different things. So back to choline. Choline by itself was kind of an unknown. It was off the wings of the performance. Nobody really knew about choline. They knew in 1850 they started looking into what lecithin was. They found out that lecithin is in an egg yolk. Lecithin it's also in liver, and lecithin is also in the brain. And so lecithin is actually from the Greek word for egg yolk, and choline is in there someplace. But that was kind of the primary ingredient for understanding choline in lecithin. And it goes on from there. I'm just going to sort of look at, so we have choline historically 1850, 1865. It was considered the mother of all substances because it is just 1865. So at the end of the Civil War, if you're an American, U.S., 1865, end of the Civil War, and it's considered the mother of all substances because it was discovered in the brain of various autopsies of humans that it was also the substance in bile. And bile is talked about. It's really interesting when you get back to Hippocrates and how he defined things as being phlegm or bile. And phlegm of the mind was blocking, too much phlegm in the mind was. And so anyways, he was talking about lecithin in the mind, lecithin in the liver. So that's where lecithin, let's say the name was derived from the Greek word for choline. It was for Greek word chol, which eventually lecithin was characterized chemically as being phosphatidylcholine. So the choline was now part of lecithin. 3 years later, in Berlin, it was another substance called neurine, entirely different. Neurine was identified in the brain. So what you had is one doctor was identifying lecithin in the brain, and he called it neurine. Another doctor, the 1865 in Berlin, identified it as lecithin in the liver, and it became the mother of all substances because it was in both places. It must be really important. It was really important. It is really important. I'm going to skip. So choline's function is part of neurotransmitters, acetylcholine. That came out in 1921. Now we're getting someplace, huh? And they called that the Vagerstoff, the Germans, for acetylcholine because it was, again, in the brain, and it was also used in muscle. Choline is a central nutrient. That's kind of what we've been talking about. But what you don't know is the people who isolated insulin from dogs that they were experimenting on at the University of Toronto in 1920s. So 1925, insulin was isolated from dogs and was given to type 1 diabetic kids, and it changed their life. Not for everybody, because they didn't know how to dose it. They gave too much to some people. But that was, let's see, it was Dr. Best. Oh yeah, Charles Best and Frederick Banting were the two that isolated the insulin. They also are the ones that went further with choline. They found that choline was found to prevent bone disease, porosis, that occurred in chickens. The effect of bone, the positive effect that choline has on bone formation we'll cover in future topics, but nobody knows about that. You've never heard anything about that, but yet choline is very much part of that. And you think of acetylcholine has a lot to do with muscles, which has a lot to do with bone. Dietary choline was found to present, yep, that was in the 40s. Acetylcholine to proving human requirement for choline. That was a long stretch. In 1977 is when Dr. Zeisel started to get involved with understanding choline up at MIT. He later became University of North Carolina. But let me go back for a second. The era from 1940s through the 50s and into the 60s, really up into the 70s, was pretty interesting. Let me just read you some of what they discovered out. I told you about the bones. That was 1940. In rats, choline deficiency was associated with atherosclerotic changes in the aorta, meaning that the arteries were not formed very well. And in the carotid arteries, so the coronary and the carotid arteries, young rats deprived of choline developed hemorrhagic kidneys and interocular hemorrhaging. Studies were published describing choline deficiency in rabbit, calf, guinea pig, hamster, baboon. And by the mid-1950s, investigators began to recognize that choline deficient animals also developed liver cancers. Let me repeat that. They discovered in the 50s, by working with these particular animals that I just named, and yeah, you had pig, and you had cow, and and guinea pig, and hamster, and baboon, and rabbit, and rat, of course, and mouse. They found that choline deficiency resulted in liver cancers. They hadn't gotten to the fatty liver part yet, and the non-alcoholic fatty liver disease, but they got to liver cancers. It's like, wow, a deficiency that causes liver cancers. Why that's interesting is because today when we talk about cancer, we tend to talk about some sort of exogenous pollutant, a toxin that got into the body and caused a cancer. Or we talk about genes like BRCA1 and BRCA2, which are the breast cancer genes that have a high penetrance for women, of course, for breast cancer. Okay. We have choline. So from the liver cancers led to interest in choline deficiency about methyl donors. So even back in the 50s, they were being aware of methylation, which is now pretty topical, right? And despite all of the animal findings and the effect of choline deficiency, scientists continued to believe that humans were different and did not need to eat choline. That remained that way all the way up until 1998, when choline was made a nutrient. So now we go from acetylcholine to proving the human requirement. I told you when that ended. And let's see, we're going to go to pediatrics. This is really pretty interesting. This is how in human studies, you had Zeisel, who was at MIT, and he's saying this is about, he was working on neonates at the time, and said they found the choline and acetylcholine made the observation that blood choline concentration were higher in neonates, right? Neonates are babies, new babies, than in adults. In part, this was because the mammary gland transports choline into milk, human milk, for the babies to have, and neonates were therefore fed a very rich source of choline from the mother's milk. Okay, this is not always the case when infants were fed commercial infant formulas. So they discovered that there was a big difference, a big ingredient missing between infant formulas that were shy of choline and breast milk, and we're assuming these were mothers that didn't have that PET SNP that I've talked about, PEMT SNP, that prevented a lot of choline creation, right? What they call de novo, making choline creation within the mother. Okay, in 2007, most commercial formulas are finally humanized. So it took from the 70s to 2007, that's about 30 years, for them to realize formulas were deficient and were causing a deficiency in children. So from there, they showed that the endogenous biosynthesis, de novo creation of choline, of phosphatidylcholine in the brain and liver was increased in newborn rodents, contributing to the very high choline concentration observed in newborns. Adding to this story was choline's importance in early life, and others were describing how choline was concentrated along the placenta to be delivered to the fetus. It seemed that the mother was designed to deliver large amounts of choline to the fetus and infants so as to sustain a very high tissue concentration of choline. So what does that mean? That means in the prenatal and postnatal formation of a human, a baby, that there's a high choline need. Why is there a high choline need? Because that's where the brain is developing, right? There's a lot of brain growth, especially then and for the next couple of years, more than a couple of years, the number of years through childhood about brain development. So there was a husband and wife team at Columbia University that was very clever, and here's what they did. They became aware of the neonate research in choline and wondered whether a high perinatal choline concentration in brain might influence brain function by increasing acetylcholine. So they thought if you gave choline to both the mother and the neonate, would they show any signs of increased mental capacity and any kind of, and they have tests for that. And sure enough, they found that out. They made the very important observation that the dietary availability of choline to the fetus, and they did it in rats first, of course, had major effects on performance in spatial maze tests. So the hippocampal function, hippocampus, right? Talked about that in videos. It's the bottom of your brain that's responsible for the learning and is responsibility for memory. So in situations like Alzheimer's, dementia, mild cognitive impairment, what is being cognitively impaired? What's being cognitively impaired and limiting one's ability to learn and one's ability to remember is the hippocampus. Okay, so this observation led to sustained effort to determine the underlying mechanism for the effects of choline. Among several potential mechanisms, they found that choline is an important epigenetic modifier. Think of methylation, changes in methylation of DNA, and that this modulated gene expression in the brain resulted in enhanced neurogenesis in the hippocampus. So what I just said is having sufficient amount of choline at the time of in vivo formation of a fetus to postnatal development of the child, that it's very important to have sufficient choline, because that's turning on and off the genes that need to be turned on. And there's a lot of genes that need to be turned on as you're forming this baby into a young child, into an older child, and into an adult. So the genes for brain development, to be simple, and there's a lot of genes, of course, need to be turned on. What is controlling that? What is modulating that? What is that epigenetic? It's choline turning on those genes. Okay. During the period of exploration of choline's role in brain development, it was discovered choline was required for so many species of animals, but was thought not to be required for humans. Duh! And let's see, a number of papers came out. And also along this time, interesting paper reported that patients fed, these are humans now, patients fed with total parenteral nutrition solutions had diminished choline capacity. So what does that mean? That means parenteral nutrition is when people go into the hospital and for whatever reason they can't eat, either because they're so weak or they can't eat through their mouth or due to injury, so they get a port put in. So you get a port put in and it has kind of pre-digested food, right? So it goes right into their blood. So it's already pre-digested, the blood. They don't need to eat anything, but they're getting nutrition. Well, these formulas were made, for whatever reason, they didn't have choline in them. Certainly this was back during the era of egg phobia, right? Don't do anything with saturated fats. That left eggs out, and that left a lot of things out. So consequently, you had these particular, now for adults, anybody who needed a port put in for parenteral nutrition weren't getting choline, and consequently we're suffering the consequences. So of these consequences, the number one consequences they found of patients that received parenteral nutrition was the fact that they developed fatty liver. Fatty liver, so how is fatty liver diagnosed? Fatty liver is diagnosed by simply an MRI. They do an MRI of the liver and go, it's larger than normal. That's kind of the obvious, and that's basically how it's diagnosed. But they also look at liver enzymes and so on. And so they found that fatty liver was a large liver that was obviously very fatty. The triglycerides couldn't get out of the liver. And they also found it had elevated liver enzymes, so something was wrong. So that came down to choline deficiency. So that's called, now it's called non-alcoholic fatty liver disease, more than just fatty liver. So in 1991, it was reported that men fed defined diets low in choline content developed fatty liver, and liver damage and resolved when choline was reinstated. In subsequent studies that included women, they found that premenopausal women often did not develop an organ dysfunction. So they go, wait a minute, we just had an experiment with men in which we deprived them of choline and they had these problems. We gave them choline and they got better. We did it to premenopausal women, and yeah, there was some problems, but most of them did fine. And so why did they do fine? Is because premenopausal women, due to their higher estrogen levels, were able to generate their own choline. So consequently, they were at far, far, far less of a risk. But they also discovered there was a number of women that couldn't manufacture, create, generate their own choline. So therefore, they were at equal risk, just like the men. So, and they identified the SNP, and I'll let you read about that because I've covered that before. And so consequently, it came down to those who don't have enough choline obviously get fatty liver, but on the way to developing fatty liver, you have liver enzymes that increase, so you're getting liver damage. And you're also getting muscle damage. And they didn't measure cerebral output, or they didn't do head shots. That would be interesting, even MRI. But we know that there obviously would be cerebral deficits in choline. There would be less acetylcholine, for one. Okay, so anyways, by 1995, the parenteral nutrition issue was resolved. It was identified, resolved, and fixed. They obviously added it in, and so that made a big difference. But there again, that's a perfect case of something was so refined, they were trying to give nutrition to somebody in the hospital, couldn't eat for themselves, and it was an incomplete nutrient meal, and consequently it suffered the damages of deficiency, specifically of choline deficiency, which are huge. It's muscle, it's liver, it's brain, it's your immune system. So what you had is all this information was put together, and finally in 1998, NIH, or the Institute of Medicine, said, okay, enough already. And that's pretty much it. It's pretty much it. And so the line of in questioning that came out of this, and so there again, I've just more or less read you a good part of the paper, and I hope you read it on your own, was that, so what is this line of liver cancer that comes out of primarily rodent and other non-human studies, that, what is the cancer and choline deficiency connection? It's not just liver. First and foremost, it is liver cancer, but it spreads to other cancers as well. So could it be that choline is an immune support thing in a whole different way, that it's kind of, I won't say anti-cancer, but if you have a deficiency of choline, you will then have a higher risk of cancer? I would think that would probably be the case. But that is so far away from today's research. So in terms of building a conclusion, brick upon brick upon brick of study upon study upon study, you can't get there. You can't say, oh yeah, it's all about cancer. The question's there. The research money has to be put up, but in my mind, it's certainly a component that has to be looked at. So I will leave you with that to read the brief history of choline. Came out in 2012 by Dr. Stephen Zeisel at the University of North Carolina. And I'm briefly going to mention another one because, and this is a little more technical, and this is called the Neuroprotective, it's a review, Neuroprotective Actions of Dietary Choline. It came out in 2017, so 5 years ago. And why would you read that? I got to give you the punchline, eh? So the punchline is a review of all the conditions that were associated with choline deficiency. You say, well, didn't we just do that in another paper? Well, this is actually 5 years more recent than this other paper, but it goes into now with more information why it is an essential nutrient for humans, what its role in development and cognitive function, that's a big deal. Also the role in epilepsy, haven't heard that, have you? Choline being involved in epilepsy, involved in Down syndrome, involved in Rett syndrome, which is a sex-linked genetic condition. So that's an interesting story. Involved in schizophrenia, involved in Alzheimer's disease. We've been talking a lot about that, so you have a leg up on that one. Involved in autism, involved in neuroprotective effects, about cognitive aging and Alzheimer's, of course, you heard that before. This is new information. And choline availability in its different forms, phosphatidylmethylamine, cognitive ability, so you'll get into that. So the conclusions are interesting because it comes up with a fourth function. This is the paper that pretty much defines and opens up the fourth function of choline. The first function, and they're co-equal, by the way, so it's not like one is better than four. They all have to be done, right? You have to be able to walk and chew gum at the same time. It's never a choice. Okay, so the number one is it's a precursor, and it's dependent. Having your choline allows you to have acetylcholine, which has to do with muscle, and it has to do with brain primarily. Has other things, but those are the two. So therefore, sarcopenia and dementia, as you get older, would be the two that have to do with choline deficiency. The second, it has to do with, I'll say, making phosphatidylcholine. Even though the form we usually take, choline, is phosphatidylcholine, it gets broken down, and then we make more phosphatidylcholine and other fats, sphingomyelin, phosphatidylcholine, and so on and so forth. But it's the wrapping of every cell. It's the wrapping of every cell. It's really important. So that was the third function. The fourth function is that phosphatidylcholine has to be available to transport in an essential fatty acid into the brain called DHA. So you have, call it a fish oil. DHA and EPA are the two initials of the two essential fatty acids that you get in fish oil. So the DHA, which makes up 90% of the essential fatty acids in the brain, and it makes up 40% of all fat in the brain, whether it's white matter or gray matter. So it's a big player. It's a big player. But in order to get that into the brain, it's dependent on phosphatidylcholine. It's like a bus. And so if you don't have any buses, it doesn't matter if you're getting fish oil, and certainly, or if you're deficient in it. And so when we see cognitive impairment, when we see dementia, when we see the brains of Alzheimer's, we see, what's with the brain? Why did it shrink? Well, it shrank, and I'm sort of giving you the crib notes, but I hope you read this, is that as you become deficient in choline, there's a hierarchy. The hierarchy is, well, we need the neurotransmitter more than we need phosphatidylcholine. And most of the choline in your body, 95%, is in the form of phosphatidylcholine because it's around every cell, right? Well, now it starts to extract it from those wrappings, cellular wrappings, to make acetylcholine. So it's kind of like a train that needs to start breaking down the wood of the train to keep the engine going. The higher priority is to keep the engine going, and therefore you sacrifice what you can. So as you start sacrificing the structure, the actual structure of the cells, the wrapping as I call it, that's the inside. You're starting to sacrifice some of that fat in the brain, and your hippocampus is the part that's responsible for your learning and memory starts to get sacrificed. So that's the fourth function of choline as the transporter for DHA, and therefore that's a really big deal. I'll leave you till next time. I think this is really exciting. I hope you do too. Hi, I just forgot to mention that, while you're listening to these different episodes, whether they're from a long time ago or something I'm doing currently each week, feel absolutely free to send me a question. Say, hey, while I was listening to you, this is what I thought. What are your takes on this? The reason I'm asking you to do that often opens up a whole avenue of thinking and thoughts, and even something I can explore for a future episode. So from a simple question to something more complicated, or explain your situation, I won't get back to you necessarily right away in a couple hours, but give it a couple days, maybe even a week or so. But I pretty much get back to everybody who has a legitimate question, and I give them a legitimate answer. And sometimes if I don't know or can't give them a very specific answer, I say, I will look into it and I'll make an episode of this. So feel free to communicate. I hope you enjoy what we're going over. This is meant to be you and me on a walk together. Kind of, I'm talking to you, but you can talk back to me should you want to. Till next time, okay? Take care. Bye-bye.